Okay, good evening, everybody. Hi. That was very positive. Lovely. Thank you for coming along this evening, especially I know there's a very sunny ball of fire out there that you could be hanging around with, so it's always nice to have you actually come in. But I think this one is going to be worth it. Okay. So it's my great pleasure to introduce Dr. Andrew Brownlow, who's come here this evening. Andrew is actually a veterinary pathologist, which is possibly the second coolest job title after penguinologist. So we're very pleased to have him here. He's actually a vet by training and then has a PhD in epidemiology, so he knows a lot about wildlife disease. And since 2009 he's been heading up the Strandings at the Scottish Marine Animal Stranding Scheme. So he has a wealth of fascinating and grotesque stories he's come on to share with you tonight. So I hope you join me in giving me a warm welcome, Dr. Andrew Brownlow.
Thanks very much, Hannah. That is far too generous a welcome. I basically walk around dragging dead things off beaches and trying to find out what happened to them. That's basically it. It's not particularly sophisticated, but thank you for coming and listening to what I will hopefully try and explain about why we do this stuff. So I thought I'd start by sort of telling you sort of why are we interested in looking at dead things? Why are we interested in strandings? What can it sort of tell us? And this is the sort of list of the sort of the normal reasons that we sort of wheel out when people sort of say that's a very strange thing you do. Why are you interested in it? And the point is that we are sort of interested in these species. The term charismatic megafauna is used a lot, but they capture the public imagination. People are interested to know what happens to animals that end up from the sea on the beach. And in specific, we're bothered about trying to understand whether or not they're there because of something that we have done as a civilization, as a species, the anthropogenic effects. So things such as the effect of toxins, chemical pollutants, underwater noise, the fact that we're very good at extracting fish from the sea and therefore there's issues with competition for prey stocks. And this sort of leads into this idea that by looking at animals that have stranded, you can somehow use these as sort of sentinels as what is going on in the ocean in general. As you all appreciate, given that this is a field that you work in, it's actually pretty difficult to understand what is going on in the oceanic environment. It is much, much easier to work on land than it is in the sea, as I'm sure you all know. So we get this data and essentially from this we can get data and samples which can be used for research, and some of this stuff is kind of slightly biased, and I'll talk a little bit about that, but some of it is stuff that you cannot get by any other means. You cannot extract this. We decided that whaling was a bad thing in the 1980s. That was a good decision that we made, I think, in general, but it allows us to get samples that we can't get by other means, certainly not in this country. So in general, the one rule about strandings is that they are always further away, harder to get to than you think that they are. I guess that's two rules, I suppose. And this is January. This is kind of a typical sort of how your year starts. Is that there is a porpoise that has stranded somewhere in Edinburgh. Now, that isn't the porpoise. That's definitely not a porpoise. So we had this. This is a sperm whale that came in. And it was, this is a problem. It sort of shows how our relationship with the marine environment has entirely changed. 50 years ago, 100 years ago, that was a resource. That was something that was really, really useful. There were people that were desperate to get hold of it for oil, for meat. It was a very positive thing to have happened. In fact, there's an act, the Fishes Royal Act, which is dating back to 1324. Which means that these animals are actually the property of the Crown because they were so valuable. Now, these days, Liz and Phil aren't that interested if you deliver a large sperm whale to Buckingham Palace. They don't want to know. So it falls to Marine Scotland and therefore it falls to us. I will try one day. So, and this is how we would do it. So the animal was sort of pulled back out to sea again and taken across the Firth to a deep water port where it was stropped and lifted out, and obviously by that time it's got dark, and there's lots of plant machinery, and we had various conversations with people that were sort of moving them, and they said, great, so we've got an 11-metre long container. I went, well, that's great. We've got a 14-metre long animal. And they went, what does that mean? They said, well, roughly speaking, it means that 3 metres of it is not going to fit. So this got taken into the lorry and then driven to a quiet part of Edinburgh before it drove through the city centre and was driven down the A1. So that kind of gets people's attention, but and we take this out, and that went to landfill, and that was where we were able to do a post-mortem. If you've ever been to a landfill site, then you'll know kind of how very strange they are. If you never have, I suggest you go. It changes how you see waste, how you behave with waste if you go to a landfill site. Other cases, this is a very old slide of when I first started, but I think this is a nice sequence. So this is a fin whale that's stranded on the beaches of Uist. Even getting out of the water was difficult enough because of the size of them. For future reference, if you ever need to use a fin whale, you need 2 16-ton swing shovels because one of them doesn't really work. So we pulled this thing out of the beach, rolled it up, and then did a post-mortem. And this is basically exactly the same process as we would use. It doesn't matter whether or not it's a fin whale or a harbour porpoise. This is sort of what we're doing. We're taking samples. We're trying to establish to see what's happened. It's a bit brutal. Sometimes you can use a small knife, sometimes you need to use a JCB. It's just a matter of scale. The process is the same. I always put this one in because I think this is just amazing. Does anyone know what that is? The heart, yeah. So this is the heart of the fin whale. And I take this out and show this for a couple of reasons. Partly it's just amazing. I mean, they are mammals. The anatomy is very similar to what we have, to a mouse, to a dog. So you can, from a pathology point of view, there's lots of similarities. In this case, the reason was actually we were able to weigh the animal because from the days of whaling, there's a good logarithmic relationship between the weight of the heart and the weight of the animal. The heart weighed 133 kilos. The animal weighed 27 tons. Standard unit of pathologists is 3.5 double-decker buses. So these are the kind of extreme cases that we were using to sort of to move around. Sometimes, in this case here, we don't actually ever manage to get the animal to make landfall. This was the Isle of Skye, which is a lovely sunny place and always. This was December. It was a force 7 blowing, and they were threatening to close the Skye Bridge. So we were on a bit of a rush to try and get this done. And this is a Cuvier's beaked whale, which had live stranded. So quite unusually, it live stranded, it had been euthanized, but then the tide had come in and it had floated off again. And we decided that we wanted to sort of try and post-mortem it as best as we could before we got marooned on the island. The reason that everyone's looking really cold is because everyone was really cold. But interestingly, we were able to do the sample collection, and we opened the stomach and found this. And this is quite unusual, but it is quite dramatic. We found 4.5 kilograms of plastic that was in the stomach of this animal. Now, this tells us two things. One is that this is hugely dramatic. Every single one of these pieces of plastic is there because of human action, or rather, through human action, we could maybe have stopped that plastic from getting into the marine environment and being ingested. However, on the flip side of it, of the 2,325 necropsies that we have done, this is the only animal that has actually died through direct plastic ingestion. So the issues with microplastics and contaminant transfer and all that stuff we can't really say, but direct plastic ingestion, what had happened in this case is a piece of this is silage wrap, had got through the stomach and then gone into basically the large intestine and caused colic. And the reason the animal stranded alive was that it was incredibly sore, I imagine, because it had essentially got a strangulated colic. So anyway, cheery note, it will press on to more death. This is a sort of also our sort of nightmare scenario, and I'm just going to talk to you a little bit more detail about this. These are mass strandings. These are the things that you really, really do not want to have in the morning. This is when lots of animals strand in the same location, and you have to try and manage this. And you can see from this picture here, there's lots going on. There are live animals involved, there are dead animals, there's a requirement to sort of try and work out what to do. This is Pitt and Weem in 2012 when they all stranded very close together. You can see how close they are on a beach, and we ended up having to pull them up that cliff face to necropsy in the field in the top. This is a mass stranding of sperm whales that happened on the island of Texel in the Netherlands, and Mario and I went out to sort of be the dialer pathologist service to go and to do some PMs on these. This is a mass stranding in 2011, which sort of kicked all this off. This is the Kyle of Durness, up on the very top of Scotland. And this is 2015, a mass stranding of pilot whales off the coast of Strathing. You'll notice these are mainly sort of pilot whales. Pilot whales are not great at being able to seemingly distinguish the land from the sea. So we do tend to get quite a lot of these species that will strand. And this sort of shows how kind of awful it can be, that you've got live animals here, you've got, as the tide's coming in, the animals that are still alive are finding it difficult to be able to breathe. You've got members of the public that are out sort of trying to keep animals afloat. And all this is happening in a big area of a very tidal sandbar in the middle of the Kyle. About 15 minutes after this footage was taken, the tide had come in so far, we were all floating, we were, into the Kyle. So that was the boat support that we had, which quite literally saved our lives here. But there was they're difficult things to sort of manage, and we try and sort of understand what happens with these. So this is where they are. There's also I haven't got any footage of it. There was a mass stranding of common dolphins down in Cornwall, but so these are the areas that have no particular real pattern with them. And when we're investigating them, this is the most extreme sort of in-depth investigation that we would do. So we look at all of these potential factors. We look at infectious disease. We look to see if there's trauma. We're looking to see if there's the effect of contaminants, algal toxins. We're looking at sort of behavioural factors, like have they got scared into a particular area by sort of, or they followed in prey, or been scared in by, for example, killer whales. Is it something to do with the climate? Is it something to do with noise in the marine environment, either natural from sonar, for example, or anthropogenic from sonar, or natural through earthquakes? And the investigation that we did, which took quite a long time, we came to a few conclusions. So the 2011 mass stranding, we found that for those of you who've been at Kyle of Durness, topographically it's this sinusoidal, highly tidal Kyle. It's a perfect whale trap. It's similar to Farewell Spit out in New Zealand, which is another hotspot for whale strandings. It drains almost entirely. Animals can get caught in there on a high tide and left quite literally high and dry. One of these animals, one of these males, had a very infected shoulder joint. So it actually had a highly pullant shoulder joint. So there's an idea of like, is this an animal that was sick? Was it sort of unable to swim properly? Had the others come into shallower water to sort of try and protect one member of the pod that was injured? And then there was the potential anthropogenic effect of the fact that the MOD had let off 3,000-pound bombs in the water just to the north of the Kyle about 24 hours prior to them all coming in. And this obviously got our attention because we wanted to try and understand if this was potentially the effect. So this is the Kyle here, and the area that they were bombing, if I can find is this little island here, Garvey Island. It's about the size of an aircraft carrier and getting smaller because they use it. It's one of the only places in Europe which they use as live fire. So it's air to ground, sea to ground, live fire ordnance. And mostly they tend to hit the island, and that's fine. That's well done. You get your badge. Sometimes that doesn't work, and the ordnance ends up in the sea. So once every year or so, some incredibly brave divers go locate the unexploded ordnance, fit basically plastic explosive to it, and detonate them, and that's what happens. So I'm a pathologist. I don't understand how noise particularly works in the oceanic environment, but I do know that it's massively complex, as probably people in here will be able to tell me far more than I can understand. But from a really, really basic point of view, this is what happens from noise on a mammalian body, be it human or whale. If you happen to be close to the source of it, you get blast trauma, and that's physical damage. That's where you can actually, the shock wave has passed through the tissues, it has caused rupture, and that is detectable pathologically. It's very easy, reasonably easy to see. We did not see that in any of the pilot whales that we looked at. A little bit further away, with a little bit less energy, you're into the realms of acoustic impairment. This is where you've affected the cells in the ear that encode sound into electrical signals, and the animals essentially can't hear. And obviously that's significant for animals that forage and feed and communicate using sound. If you're essentially rendering them blind. And then at a much greater area, you've got the behavioural disturbance. You've got the responses to just simply not liking the noise, not liking the novelty of it, the effects that has on feeding, on migration, etc., etc., the physiological stress. Now, the problem with that is that leaves no pathological legacy. There's nothing to detect from that. You can't tell that an animal has obviously come in there because it was scared. So this is what we were faced with. This is where we were trying to work out how successively we could understand what happened with this sort of acoustic impairment. So if we scroll forward from 2011 up to 2015, this is the mass stranding that happened on Staffin Island, and in this case we were very careful. This is actually also 2012 to get the brain and the ears out of these animals as quickly as we possibly could. We've got a very small window of time before we can no longer distinguish the effects that have happened due to sound from the effects that have happened due to autolysis. So we've got about between 7 and 20 hours in order to be able to do this. So this kind of involves turning up, and as you can tell, that's at night, taking the brain and taking the ears out. This is the ear of a pilot whale, bony structure that's buried inside the head, and this is us fixing it with a formal saline. So you take the ear out, you inject it with formal saline, and we send it to an amazing colleague of ours, Maria Morel, who at the time was working at the University of British Columbia, and she had made these absolutely beautiful sort of electron micrographs. So this is the Cochlear Spiral. This is the part of the ear that codes the sound into electrical signals. And if we zoom into this area more and more, so this bit here is a cross section through that spiral, and taking a cross section of that, this is where the actual process happens. These are the hair cells, and they are the cells that actually do the process of taking sound that moves the tectorial membrane and turning it into electrical signals. Now, the important thing is that if these cells get too loud a noise on them, they cease to work, and if it's significantly too loud a noise, they burst and they die. So if you've ever been exposed to a very loud percussion noise, like a shotgun going off, for example, and you've not got earmuffs on, and you end up with that high-pitched ringing in your ears, that ringing is those cells dying. You will never ever hear again at that very specific frequency that was involved. So what you have here is a way of being able to image these cells to try and see if these animals potentially had hearing. And we did this. We looked at the normal architecture. So this is a normal bit of the ear, and you can see these lovely horseshoe-shaped things are the outer hair cells. That's how they should be. That's how they should look. And from that bit of the ear, we could be reasonably confident that this animal was able to hear. And then in some regions we had this. You can see that there's a total loss of these cells. So, I mean, that shows that there was probably no functional hearing at the frequencies that were coded for by this. Now that's great. Does that mean that we now understand that this was done by exposure to loud noise? No. What we can't do at the moment is work out how long it takes that to happen. We don't yet know whether or not this takes 2 hours, 2 weeks, 2 months. We have no idea. But it is a way, it's a start for us being able to look at the effect of underwater noise on these cetaceans. If we can get to them in enough time and we can fix them in this way, we at least now have a tool to be able to do this. And obviously we got the Guardian slightly took the wrong end of the stick when we said we have no idea what caused this, and they went, the Royal Navy bomb caused the mass whale deaths. So this is a slight issue with PR here, but we've got a paper here about this, so if you want to know a bit more about it, this is kind of what we did from all of that. And finally, this is on Monday, or rather this animal at the bottom was from Monday. This is with these sort of mass stranding events, this rather amazing footage at the top was taken just off White Ness, I think it is, and you can see there's a pod of pilot whales here that are doing collaborative feeding, all diving at the same point, and then a few days later this solitary aged female stranded on the beach just outside Inverness. And we had a really tough decision to make, because the rest of the pod were well up past the bridge. They were in the Bewley Firth, which for deep diving species is the wrong place really for them to be. And we were very concerned that this potentially meant that if this animal went back out to sea, she could potentially bring the other ones in and cause another mass stranding. And as I said, you know, there's 2 film crews watching us there. There's live and dead animals potentially involved in the mass stranding. And in this case, I decided that this animal needed to be euthanized. And that is a really tough call to have to make. Now, we took it to post-mortem. We were able to take all these samples that we needed. And in this case, she was a very old animal. So it is possible, I mean, this is all conjecture, but it is possible that this animal wasn't really being able to sort of navigate quite as well. She'd got so much white matter in the brain that she was almost, you know, it was almost like she'd sort of got, it's very similar to what you see with sort of people with Alzheimer's or dementia. So, I mean, we'll find out from the histopathology, we don't know this yet, but this idea of the sort of sick leader, if we removed her from the equation, would the others go back out to sea? And so far, touch wood, the others did go out and we haven't seen them since. So hopefully that was the right call. Okay, so that's the acoustic bit. Has anybody got any questions about that they want to ask just at this point? Because I'm going to go and talk about something totally different now. Nope, good.
I've just got a quick one. If you're calling the JCB, who picks up the tab of that?
Oh, interesting. Now that depends. If it is 25 feet or greater, then that's the Scottish government, because that's the royal fish. So local councils are really keen to make sure that animals are 25 feet, because then they can bill the Scottish government. Under that, in Scotland, it's the responsibility of the local council to do that. And that's why our job is sometimes we provide a disposal service in some way. We will take them away and at least be able to sort of make sure that the problem is contained, because unsurprisingly people aren't tending to be used to what happens when they get a large whale on their doorstep. Yeah, no problem. Okay, so I'm going to talk about something else there. So that the mass stranding stuff is about how we sort of try and work out what effects you have on an individual population. This is a different story. This is about what we can learn about the marine environment from single individuals. Obviously this is a killer whale. This is a very thin animal that's stranded out in the Uists, and from this we can take blubber samples. Now the blubber here is incredibly useful because we can analyze this to look for certain contaminants, and because of this what we know about killer whales, we were very interested to do this sort of type of analysis. And then spin forward a couple of years, this is another killer whale. This has been essentially the poster girl for human impact on killer whale populations. This is Lulu. She was a member of the West Coast population. She stranded in early 2016 on the Isle of Tiree. And the reason that she stranded, the reason that she died is because of this. These are entanglement marks, chronic entanglement marks. Her tail has got wrapped around a rope, possibly, most likely the sort of rope that goes from the sinker buoy from a creel float up to the surface. And she's become entangled and she's drowned. So we went out, we post-mortemed her, we established that that was the case, and then we sort of looked at some other potential reasons of what was going on there. And because killer whales very, very seldom strand, and they're quite difficult to get tissue samples from. We do quite a lot with other collaborators, work with these sort of samples. So we've sent some skin, tiny bits of skin off for genotyping analysis, and found that so far she was homozygous for all 9 markers that were used, which shows that that particular population is very, very, very inbred, and there was no mixing with any of the other populations that exist from the Iceland or Norwegian populations, at least that we could tell. We're able to age the teeth, and I've got, later on I can show you a sperm whale teeth for how we do this. You can cut the teeth longitudinally and count the rings exactly like you do in dendrochronology. It's the same process. You can age animals, age cetaceans through teeth rings. So we did the teeth age. You can find that she was at least 20 years old, but probably was quite a lot older than that based on her skeleton. And what was then interesting is that we looked at her ovaries and found that despite the fact that she was well past the age of sexual maturity, she had no mature follicles or corpora visible, so the ovaries were essentially inactive. And essentially, it looked like she had never had a calf, or never successfully taken a calf to the point at which it had implanted and was viable. And what I mean by this is, these are the ovaries of Lulu, and if you cut them in cross section, you can see that they're just kind of fibrous. There's nothing really sort of interesting, no particular structure in there. Compare these to ovaries from some killer whales that stranded in Japan that were much healthier, and these nodules here, these are the corpora, and you know it's pretty kind of bucket science here. You just cut them and sort of count these. These are from animals that have previously had a corpus luteum, which is then turned into a corpus albicans. So this shows that they were reproductively active, and that was not the case with Lulu. And one of the reasons that we think this could possibly be is looking at pollutant burdens. And one of the particular pollutants that we were interested in looking at were the effect of PCBs. So this kind of chloropleth map here shows animals that have been screened from around the UK for their PCB burden, with green being relatively low, yellow being kind of not great, and red being a bit dodgy. And you can see there's sort of certain areas around Wales where there was incidentally a PCB plant that was about there, I think, so I'm not sure what that potentially means. But the north coast and the west coast, potentially something going on. Now, a quick kind of explanation about PCBs. They're basically 2 benzene rings that are bolted together. And around the outside of them, in all of those different positions, you have chlorine atoms that sort of stick there. And they're very small molecules, very planar molecules. And as a result of that, they move through tissue very easily. And they also can act as sort of hormone analogues. So they can confuse the body into thinking that they're cell signaling molecules. And as a result, if you have PCBs in your system, then your normal body cell signaling systems don't seem to work that well. So you have a whole load of problems. You know, they are mutagenic, can suppress the immune system, and as I said, they're hormone analogues. They're bad news. We know they're bad news. You know, it's taken us quite a while to sort of to realise this, but they were banned in the 1980s because they're known to be toxic. Specifically, they bioamplify and bioaccumulate up through the food chain because they're very lipid soluble, because they're small benzene rings stuck together. They move up through fatty tissue. So if you have animals that feed near or top of the food chain, that's where you're going to end up with them. And what we were looking at was sort of the levels at which we think that PCBs are sort of a problem. And it's a bit kind of questionable about these thresholds, but these are ones that we chose. So this here is your PCBs. Well, the more PCBs you've got, the worse it is. These are looking at Harbour porpoise, and these 2 lines drawn over here. The bottom one, the yellow one, is what's called a Cannon threshold. That's the sort of the level in experimental conditions where you're beginning to see an effect. It's about 11 milligrams per kilo, 9 to 11 milligrams per kilo. That's when you're beginning to get physiological effects. And the black line is slightly higher. That's in the 40s. That's a little more arbitrary, but that's the level where we're pretty certain that they are causing physiological effect. And you can see that Harbour porpoise here are sort of well under the 40 level. Some are slightly over the 11. Interestingly, you probably can't see this, but the ones that are the highest are male harbour porpoises, which have died from an infectious disease process. So there's a bit of a question there about maybe there's something going on here. Those are the killer whales that we've looked at, and these are the means. So well over the threshold, absolutely well over the threshold. Now these are means, but Lulu was 957 milligrams per kilo. So on this graph, she's probably on the next floor, somewhere up there. She was the highest burden that we've detected in the killer whale in the Eastern Atlantic, and suggests that this particular population possibly contain animals that are some of the most contaminated on the planet. And the person to talk to about this is my colleague Paul Jepson, who has put a paper out of it on PCB pollution, and he's pretty evangelical about this. It's definitely worth having a conversation about it. He is firmly believed that this issue, accumulation of PCB pollutants in the marine environment, is the single biggest hazard to our marine cetacean stocks. Looking at this detail shows another couple of things, though, that's sort of worth just having a look at. When you say all of that, there's usually somebody, especially if you say this in Shetland, that goes, hang on a minute, the killer whales in Shetland are doing really nicely, thank you. They're breeding, they're reproducing nicely, they're doing really well. And that's really fascinating, because if you look at the data that we've got, and we don't have many of this is the problem with the stranding stuff. This sample size is tiny. This is a sample size of about 8 animals. But the ones that we do have, the Northern Scotland and Orcadian population, this is the animals that spend their time feeding on herring off Iceland and Norway. These have relatively low levels, with females being lower than males. I mean, they're still above the threshold, but you know, this is mean, there's a bit lower. The reason there's a difference between male and females is that females can offload PCBs in their milk, which is good news for them, bad news for their calf, because the milk is incredibly lipid rich and the PCBs dissolve in this. You look at the Hebrides and we've got much higher levels and pretty much the same levels in there. So the question is whether or not this represents populations that are probably feeding higher up the trophic level, so they're getting exposed to more, or they're just not managing to offload it. And I suspect it's probably that these are the much more marine mammal eaters. So they are, you know, they are the sentinels. They're the absolute sentinels of the sea. Does it mean that our seas are massively polluted with PCBs? No. It means that these animals that take the advantage of feeding on things, that feed on things, that feed on things right at the bottom of the web, they're the ones that are going to see the effect. Okay, right. I'm now going to talk to you about harbour porpoise and bottlenose dolphins. This is normally what we spend our time doing. So everything I've spoken to you about up to now is kind of the weird and the wonderful. This is the animal that we spend most of our time doing post-mortems on. This is your good old-fashioned harbour porpoise, and this is the most common cause of death that we see in harbour porpoise. These, you probably know what this is and where we're going with this, but bear with me. These were identified by my colleague and by Ben Wilson about 20 years ago nearly now, as being, initially they thought that they were to do with interaction with fishing vessels. Animals were coming in showing a huge amount of trauma. They had these parallel rake marks on them. These rake marks were associated with severe amounts of subcuticular trauma. You can see the bruising there, and the ribs are often massively broken. There is a huge amount, something has hit this animal really, really very hard. And for a long time, there was a thought that this was to do with some form of human interaction, some form of interaction with fisheries. And it was only when my predecessor, Harry and Ben, sort of went and looked at these in a lot more detail and saw that the one thing that was in common was the distance between the rake marks was between 11 and 13 millimeters. And that corresponds really very nicely to the interdental space of a sympatric species in these waters, which is the bottlenose dolphin. And at that point, the idea that the bottlenose dolphin could be beating up your harbour porpoise went down really, really badly. They had a hell of a time when they were sort of trying to get this through the ECS in 1994. And then this footage, which you've probably seen, came out, which sort of put the nail in the coffin of that, showing bottlenose beating up harbour porpoise and booting them out of the water like this. And this, I mean, this is without a doubt the single most common cause of death of harbour porpoise that we see in Scotland. It's not bycatch, it's bottlenose dolphin attack. And that's kind of interesting because we thought, okay, so that's something we thought was anthropogenic and actually turned out not to be. And if we'd have paid attention to that, we'd have probably learnt when a few years ago we started seeing these. And these are seals that were coming in with curvilinear lesions around their body, starting at the head end, very, very smooth edges to the wounds. And we looked at all the possible options of what it could be. People were thinking that it was potentially to do with people going out with knives and attacking them. There was an idea that they were sharks, Greenland sharks, and the idea that we settled on, that we all settled on, myself included, was that it was something to do with interaction with boats and boat propellers. And this was backed up by some work that was done by the Sea Mammal Research Unit, where they did some modelling, not computer modelling, real modelling with plasticine, and put this is a plasticine, wax-covered plasticine model of a seal going through a ducted propeller. And the idea is this here is a duct. You can't see it because it's clear, but essentially it's a ring, the propeller rings around the inside of it. And we thought, well, that's it, you know. So we did the modelling and thought, right, that's probably it. That's what's doing it. It must be boats, must be boat interactions. And that was the lesion that came up there. We obviously didn't try this with real seals because the paperwork's difficult. And that was fine right until a totally serendipitous accident, if you like, on the Isle of May, which has been monitored pretty closely for the best part of the last 20 years. A student was just happened to be out there filming some behaviour on a grey seal colony when she happened to notice that there was this adult grey seal male went and captured a grey seal pup and grabbed it by the scruff of the neck, dragged it about 40 metres to this freshwater pool where it proceeded to drown it and then bite at the skin at the back of the neck and essentially eat it. And that's pretty horrific. This was new. The idea of cannibalism was new. It's like, that's really strange. We've not really seen that much before. But what was more surprising was that when the carcass was recovered, it looked like that. And they had, you know, evidence of sort of these continual spiral lesions. And when we went back to look at the original photos that we'd had and the data that we had, we discovered that the mistake that we'd made was that these sort of marks here, which I'd passed off as being scavenger marks done by birds, because a lot of these cases that come in are ones that were had been subsequently scavenged by birds, actually probably weren't. And what they were primary predation. And you can see there's the holes there and there's the undermining of the blubber. And subsequently, when we've had a lot more cases in, because this is when they were just starting, we've been able to look at them in a lot more detail and have found that actually the most likely cause of death of these cases, of the grey seal weaners and potentially harbour seals, is this grey seal predation effect. And if we just look at the numbers here, so when we started off, this was the total number of potential cases that we thought were to do with what we call corkscrew seals, but grey seal attack in 2012 and 2013, 2014, 2015, last year, and then that's up to, I think, April in 2017. So there's now a question here, is like, is this because populations are increasing? Is this because we're noticing it and we're seeing it and it's something that's new? Or is it a question that in actual fact grey seals are learning that this is a new food source? And we don't know, but it's something we're working on. So I'll just kind of come to an end about this and sort of push the ball back, kind of, if you like, to you in some ways. In order to do all of this, in order to do this sort of work, as I said at the beginning, it's all opportunistic surveillance. There's three of us that run the Stranding scheme, essentially. We can't go and wander the beaches ourselves, so we rely on members of the public and volunteers to be able to do this stuff. And the information that we need is pretty basic. We kind of need to know where things are, when things were, and kind of what they were. There's a load of other stuff that we need as well, like size, condition, sex, body condition, whatever. But everybody now has the most amazing piece of scientific equipment in their pocket, and it's the mobile phone. I mean, it is astonishing. It has a GPS transmitter, and you can communicate in almost real time to people. You can send photos, you can video footage. It is absolutely astonishing. It revolutionized how we do what we do. So if you do come across a stranding once you're out there, please take some photos of it. Photos of the side so we can see what it is, photos of the genital region so we can get a sex of the animal. Scale is really important. Beaches, marine environments are fractal areas. It's difficult to work out the scale of stuff. And we use whatever you can. This is a plastic bucket next to a seal. It's very useful. If you're involved with refloats of pilot whales, which you may be, because as I said, they're land, sea, not great, understand this, please take photos of the dorsal fin, because if each animal probably has a slightly different pattern to the dorsal fin and probably has different nick patterns off the trailing edge of it, it's a fingerprint. And it means that we can identify if these are animals that have gone back out to sea again and have come in again, because the prognosis of an animal that has been refloated once and has restranded is massively different and much poorer than an animal that has stranded just for the first time. So please, if everything's going to chaos during a mass stranding event, please just take some photos of the dorsal fin. This is my favourite side. I love this. In the days before mobile phones and whatever, we used to get letters through, and one letter that turned up had nothing in it apart from this photograph, which just said on the back, bones, see rabbit for scale. Genius. To be fair, it was bones. We didn't really need to know about it, but yes, I absolutely love that. Favourite bit of it. If you are interested enough to kind of be involved with us and want to become one of our volunteers, our stranding volunteers, of which these are the distribution of them, particularly if you happen to live out on Stornoway at the moment, because we're not good at clearing this, then get in touch as well. What we sort of do is you come and we give you a day training course in how to take samples, how to keep yourself safe. That's kind of important. This is in Aberdeen University. You get to wear some natty blue equipment. You get to see, take samples from the Harbour porpoise usually, and also see me do a necropsy as well for those that are interested, and sort of explain a little bit about kind of what we do. And then we'll ask you to maybe go out and take samples for us or take photographs. And it is really useful. It is the best sort of initiative that we've kind of come up with for years, really. It's really helped us being able to get information about cases that we can't get to, because in some cases we can't. You know, it's far too remote, or in some cases the carcass is a bit too decomposed to make picking it up and doing a full post-mortem worthwhile. So what we do is just ask you to take kind of photos, a bit of blubber, which can go for toxicology analysis, some skin, which can be used for genetics and what have you. We obviously have a Facebook page, but you know, find us on that. There's updates on there that show us, you know, what we've been up to, how we've been spending Marine Scotland's money. We have a web page as well, which allows you to get lots of information. I'm an epidemiologist by training, and I love data. So there's lots of kind of bits of data up here of how you can sort things by cause of death, by where it's stranded, by when it's stranded. It uses a Google API sort of map. You've got these little balloons. You can zoom in. Obviously, you can click on a different balloon. It will tell you if we've done anything about it. You get a little report that shows what we've done. This is also getting slightly updated. We're trying to sort of make it a little more interactive. We're not quite done that yet, but this is happening. So the data is there. Main point is the data is collected. It needs to be used. You know, it is available. It's publicly available. It is free to use. Please think about using it, because otherwise there's no real point in us going out and doing all of this stuff unless it can be useful to people. So bear it in mind. If you have some ideas of some projects, you have some ideas of things that you want to do with it, then please get in touch with us, because it's there. It's there for you. So that's kind of about it. Hopefully what I've maybe shown you through in between the sort of the car crash cases and the sperm whales in the back of a truck is that by looking at these dead stranded cases, we can get some idea about the health of and threats to the population. It's not ideal, there are biases, but it is potentially useful. It helps us, the phrase I like to use is, it helps us to understand not just how animals died, but also how they lived, about what was going on in that environment. And you know, we think we're getting to the point where we can begin to look, identify and quantify the anthropogenic impact of different sort of causes of death. And the findings from that were not exactly what we expected. In some cases, some apparently HI cases, human interaction cases, sorry for the acronym, traumatic cases, have been discovered to be due to interaction with sympatric species, so they are natural. What we thought was interaction with fisheries in the bottlenose dolphin case, what we thought were interaction with boat propellers in the seal case, they actually both turned out to be to do with predation from other species. So that's good. That kind of gets us slightly off the hook. But then we go fully back on the hook again when we look at the effect of persistent pollutants. You know, we banned them in the 1980s, but we can't forget about them. They are still in the marine environment. Rather scarily, most of them are actually not in the marine environment. They are still on land. 80% of them, by estimates, because we don't really know, are still on land. If we don't watch what happens and are mindful about these sort of pollutants, they will move into the marine environment, and it won't just be these apex predators and killer whales that are having this effect. It will potentially spread. So we need to be mindful of the effect that we're having. Just because it's out of sight, it really shouldn't be out of mind. And noise is hugely difficult to kind of get a handle on, but we've got some tools there. And the thing that we're trying to work on, and if you have ideas of how to do this, please get in touch, but this idea of chronic stressors. So it's not just the bang explosion that causes animals to strand. It's like animals that have been stressed over a long period of time because of potential anthropogenic and natural effects. Cumulative stressors, that cumulative stressors is probably the thing we need to know. But as I say, the data is opportunistic, and it relies on accurate reporting of cases. So the most important part of all of this are people like you to get this. And that's all I'm going to say. So thank you very much indeed. These are all the people that help us do this. If your logo is up there, it might be old. So apologies. If your logo is not up there and you help us, I am sorry. Please give us a logo. But that's it. Any questions, please ask. Thank you.
Okay, well, thank you to Andrew, and good luck to those of you that haven't had dinner before you came tonight. We've got a few minutes if you've got some questions for Andrew.
Yeah, what do we know about levels of PCBs in fish stock?
They're quite low. CFAS actually do look at these, and I don't have the figures to hand, but I think they have been monitoring them and actually find that they are below the levels which are deemed under any metric to be problematic. What I have heard anecdotally is that they tend to look at species that don't enter into the human food chain in case they happen to find high levels. So I have heard, again anecdotally, that they don't look at place, but they do look at DAB, because the place, they feed in exactly the same trophic level, but it's easier for them. That's probably a question to ask CFAS, but they are monitored. That's the point. They are looked at there.
So where do you think the sink of this pollutant is then?
Of the PCBs, well, killer whales is a very good sink. If you want to pick them all up, some of them will go into the killer whales. In general, they get atmospherically resorted. So PCBs is not a single compound; it's an entire family of compounds, and dependent on let's say those two benzene rings bolted together and the chlorine atoms that are around there, dependent on where they are, they have different metabolic functions. So some of them are metabolized as they go through the body, and some of them aren't. Some of them are volatile and can be sorted by atmospheric recirculation. So one of the problems is that the PCB levels in animals in polar regions are much higher than we expected, and that's not because we don't think that's because there's a PCB source in the polar regions. It's because they're getting moved up by the various environmental sources. In general, what we hope happens is that they stay in at low levels, they sink and then essentially are interred in ocean sediments. But they are massively persistent. They will hang around for quite a long period of time. And the problem is that if you have if you have areas that do dredging, so a lot of the eastern seaboard where they have harbours that maybe had a PCB plant around there, when they dredge that harbour to be able to move the sediment in there. In some cases, that material actually has to go onto land and is buried. And in some cases, they have to go into land and are incinerated because the PCB level in it is so high. Now, the problem is that the states, bizarrely, I say bizarrely, that's an unkind thing to say, but they're a lot further ahead with the PCB legislation than we are over here, because they have sort of Superfund sites which have used money generated for the last 30 odd years to help with decontamination of these areas. And we don't have this in Europe. And the other problem that we have over here is that we have, 8, 9, 10, 11 different countries that all have to interact in order to work out who is responsible for what's in the marine environment, because the PCBs in this killer whale, they may have come from a UK plant, they may have come from a French plant, they may have even come from a Russian plant, we don't know. So it's difficult in Europe, but it is still something that needs kind of looking at, otherwise all levels are going to go up. Okay.
You're hinting that the interspecies aggression is a new phenomenon, and it might be something to do with competition in the food chain or whatever. Presumably history would tell us whether this is new behaviour.
Yes, this is the interesting thing, is that, to the grey seal thing, for example, we don't know whether or not the difference that we've seen from 2012 up to now is because we're looking, or because grey seal numbers have increased by about 10 to 12% per year in general. We don't know if it's because grey seal, the actual number of grey seals out there isn't the highest that it's now ever been since sort of geological, since the North Sea wasn't a sea, really. We don't know that, but there does seem to be an increase in incidence. And this is kind of, I've not really spoken about this in this talk, but this is the problem with the stranding stuff, is that we don't know if what we're seeing is to do with biology, as in something that we're interested in, or if it's to do with the reporting of strandings, or strandings ending up on the beach. And it's a piece of work that we're kind of trying to disentangle, but it might be that we're just seeing an increase in reporting cases because everyone knows about it, social media is great, all of that stuff. Or it might be because it's an effect of biology. But we'll come back in a few years and we might have an answer. Okay.
Just wondering about the acoustic, the noise damage in the waves. Have we got any other species that you can say you can repeat that pattern where you can see the damage? One can apply that to the exposure over time, or is it?
Yeah.
You were suggesting that you're looking at longer exposure, chronic exposure.
Yes, we can. It's been done in humans. It's been done in rats quite a lot. You can look at the changes over time, and specifically in rat hair cells, you can look at both the cells and you can look at the footprint, if you like, of those cells in the tectoral membrane. So this is a bit of development on the work that I've just presented here, where that allows you some idea about whether or not these lesions are really, really acute, as in within 4 or 5 days, or are much more chronic in duration. So yes, there is stuff that we can do with this. The problem is, for these particular cases, is that we don't yet have a frequency map of the pilot whale audiogram, essentially. We don't, that's the sort of the 101 basis that we really need. We need to know at what frequencies they can hear, because if you imagine the case that I showed with the attenuation of hair cells, that was actually at one very end of the audiogram. So a bit like us, I can't hear now frequencies that I could hear when I was a teenager, but that doesn't matter. I can communicate perfectly fine with you. If I'd lost that same amount of hair cells, but a couple of octaves further down, I would be essentially functionally deaf, probably. So it's not just a question of the hair cell loss, it's where in the cochlea it happens to be and what that means in terms of coding. So there's a lot of work that can be done, but this was more just to demonstrate that it is a tool that we can potentially use to start doing this. And also why that explains when you get a pilot whale stranded on the beach, I instantly try and chop the head off and start sawing at the ears, because there is some science in there, trust me, there is a reason for it. Okay. Hmm.
I was interested in what you're saying about the reporting rates. Can you give us an idea of the actual numbers of species just to kind of picture the numbers of species you've seen? And if you look at the kind of map of stranding data you've got, do you think that's representative of the populations that are out there in different parts of the coast? Or is it representative of the population of people that walk beaches and call you when they see a porpoise?
Is the stranding sort of representative of what's at sea? Or yeah.
Are you getting a picture of the live populations, or are you just getting a picture of where people are walking?
I think that's exactly the problem. We in certain areas that there is a taxonomic prejudice, as my colleague would say, about this. People will walk past a seal on a beach and they will report in a harbour porpoise. It is unlikely that we will get a fin whale that strands around Scotland that we don't know about. But the idea of a grey seal or harbour seal stranding, that in Orkney, where most of these populations are, until we started the volunteer network and really went and basically talked to them about this, the only people that would report something in were people who just moved there or were there on holiday. Because it's like, oh, there's a dead seal on the beach, and then 2 weeks later, like, oh, there's another dead seal on the beach, walk on. And so, you know, there is a reporting, there's a species kind of issue there as well. Strandings, I mean, I haven't really spoken about this. They are biased. They're biased towards things that float. They're biased towards things that are coastal. They're biased towards things that are noticed by people. So, can we use strandings to infer what's actually out at sea? That's a bit of a dodgy question. What we can do is if we find something on the beach and it's not in a particularly autolised state, it probably was alive in the sea relatively close. We had a beluga whale stranding that was pretty skeletal, but not that skeletal, off Lulu Bay a couple of years ago. That's the first time that we've seen a case of belugas since the 1930s have been reported. And it potentially means that this animal was alive somewhere off the coast of Scotland. It is possible that the carcass drifted all the way from the north, but it is possible that this animal was alive around here. So in terms of understanding presence, yes. In terms of understanding prevalence, oh, that's come back in a couple of years. Well, when she's done a PhD on it, that will work.
Okay, so we'll be back in about 4 years' time.
Yeah.
Can I just have one more question from you, Scott? Coming back to the Bottlenose Dolphin Harbour porpoise, is there a behavioural reason? Are they trying to get rid of conspecifics, or are they treating with beach balls? What's the kind of reason for these?
I don't know. I think the risk of sort of saying they're just bad, you know, they're just evil. Their flipper is not generous. I don't know. I mean, you get a slightly more sensible answer. We see infanticide as well, so it's not just a harbour porpoise. There are young bottlenose dolphin calves will get similar lesions. So is it kind of the same idea as you get with other species, with lions, for example, where they try and kill the youngsters? Probably not, because in these species there's a chance of them bringing them back into ovulation, and that's not the case in cetaceans. Is it deferred feeding? Is it prey competition? Are they smart enough to realise that they're going for the same prey? Well, I've seen bottlenose that do this. They're eating salmon that are about the size of a harbour porpoise. So probably not. But then there's the next question: is it just that they're mistaken for it? Well, I don't think so. I mean, bottlenose dolphins are demonstrably quite clever. I think it's play. I think there is a big element of this, because we see two different types of lesions. We have the ones which don't look at all like they've been hit by a bottlenose dolphin, but inside they've got sort of a single blow has killed the animal outright, and then it's not then played with like a cat does with a mouse in that sense. They've got bored and they've swum off because it's no longer providing them with any entertainment. And then you see ones that really have kind of been cat and moused, and they've been kind of lots of rake marks, and these animals sometimes even strand alive, so they've escaped. And we see that as well. We see animals that have got away, that have healed rake marks, whatever. So no, I think I'm just my money is simply on that flipper's not very nice. I think that's the.
Could I just make an observation on that? I've been fortunate enough to tried tour boats through the Cochlear Breck
Yeah.
over the last 16 years. And if we're seeing bottlenose dolphin, we very rarely see porpoise. They have the sense, I think, to get out of the way.
Yeah. That's interesting. I would get out of the way from bottlenose.
Yeah, no, but anyway, just
Yeah.
an observation.
Yeah.
Okay, well, that's a great note to end on.
Yeah.
cheerful thoughts.
Yeah.
So I'm sure Andrew's going to hang around for a little bit afterwards if you have any more questions, and feel free to stay for a drink or anything. Otherwise, I just want to say thank you very much again.
Yeah.
Thank you.